So we're going to move on. Sorry, we're a little behind time, but we're going to move straight into session 2. And this is a session that's going to be featuring some of our clinical trial readiness initiatives. I will be speaking about our RareX data collection program. Dr. Wendy Chung is going to join us virtually, and she's going to give us an update on the CACNA1A natural history study. And then Dr. Terry Jo Bichelle from Combined Brain is going to be speaking about our biomarker initiatives, which some of you have been donating blood towards. And so she'll talk a little bit about why that's important and what we're doing with that information. Okay, so I'm going to go ahead and kick it off. Welcome, everybody. Hello to all the virtual attendees out there. It's great to be back in front of you after 2 years. So I'm excited to kick off this session about some of these clinical trial readiness activities that we've been involved in. Again, I'll focus on the data from our data collection program by RareX. But before we get into it, I really wanted to just give everybody a better idea of what we mean by these clinical trial readiness initiatives, right? Yesterday, one of our researchers said that's a lot to cover in 3 words, and it may sound like jargon, but it's a lot of things that we have to get done as a community to get ready for, moving a treatment into the clinical trials, and then hopefully get that approved by the FDA. All right, so you often hear us talking about, So the main mission of the foundation is to find better and specific treatments for our CACNA1A community. And when we look at this pathway to clinical trials, meaning that a candidate treatment is formally tested in members of our community, there are a lot of steps. And this requires a lot of people, a ton of work, and then, of course, money. But together, we're going to get there. So we start off with I'm going to move back and try to get. So we start with our disease community or our rare disease, and we start building what we call preclinical toolbox, which you've probably heard us talk about a lot. We start understanding those disease mechanisms, which is what Jen had spoken about earlier, right? How does a variant impact that protein function, and how does it actually lead to those symptoms and those disorders that we see? And then we get into, that therapeutic development and those proof of concept studies. And these are those really crucial experiments that have to be done that will show that whatever treatment that we're testing works on our gene in these models. And if we can show that and we show that it's safe, then we can go towards what we're calling this IND filing. And this is an investigative new drug application that we file with the FDA. And they take a look at that. They look at the safety data. They look to make sure that it is, again, working in our models, safe in the models. And if that gets approved, we can move into those clinical trials and start testing that on our patient or our community. So you often hear us talking about basic research and translational research. And so basic research really builds that foundational knowledge of our disease. And it's all about building that preclinical toolbox. Again, these are the tools that our researchers are going to be using to start developing those treatments. And so we build our translational research based off of what we learned from the basic research. And translational research just means that we take the basic research and we apply it to people. And so both steps are really important. We can't get to the translational research without the basic research, but it's the translational research that moves us towards those clinical trials. And I want you guys all to remember this, right? Because I know you've filled out all these surveys. I'm filling those out too, but it's patient data that drives the research forward. It really is. And again, this session is really going to focus on, how we've been building our preclinical toolbox to support research. We have our disease models. Oops, sorry about that are shown here. So we have these special stem cells that have been derived, or that have been derived from patient samples, and those can be used to study this disease outside of the body. Then we also have these animal models. There's the mouse, fruit fly, zebrafish, nematodes, or worms. Those are all, again, really helpful in understanding how this disease or how these variants impact protein function outside of the human body, because we don't really want to be doing experiments on us, right? And then it's these clinical trial readiness initiatives. And basically, this is collecting data from those who are living with this disease, right? And that is so crucial. Yes, we can study it outside of the human body, but we also need to understand what is happening to the human body and who better to ask, right? all of you out there. And so, as I said, I will be talking about our data collection program that's being powered by RareX. Dr. Chung is going to be talking about the natural history study, and then again, Terry Jo will be talking about the Combined Brain Biomarker Project. Okay, so just a little bit of a refresher here. What is RareX? RareX is a platform that was built specifically for collecting and sharing rare disease data. And it was launched in 2021, and the CACNA1A Foundation was actually one of the first patient advocacy organizations that had the opportunity to join, and so we really couldn't pass it up. It's a virtual study consisting of standardized surveys that aren't actually specific to CACNA1A, but that's actually okay, because it can help us find overlapping symptoms between all these different diseases. So if there's a researcher who's working on a drug for ataxia, they can go into RareX and they can search for ataxia, and they'll see some of our community in there. And so that's an example of why this is important. It can also help us find symptoms that are maybe underreported, things that maybe we don't always associate with CACNA1A related disorders, but it is there. And another point too is that this data isn't exposed to, again, this more global audience, and RareX offers these great initiatives like the Open Science Data Challenge, which happened last summer, where a ton of researchers from around the world were able to actually get a hold of our data and study it. Unfortunately, whatever their projects were, they weren't funded because it was sort of this contest. But RareX is going to be holding these types of initiatives again, and so hopefully somebody will take an interest in our data and move forward with that. And I also want to emphasize that this is not meant to take the place of any other data collection program we're doing. It's not meant to take the place of the natural history study. This was built to complement our natural history study because it's a more broader application, and then the natural history study can really focus on what's happening within the CACNA1A community. So here are some of our enrollment metrics in terms of who's participating, where they are. So right now we have 125 participants enrolled, and this map is showing you the distribution of where everybody is. You can see most of the, oops, sorry, I keep pressing the wrong button for the laser pointer. You can see most of the participants are in the US, but we still have a global distribution there, so that's great. We have a 49% completion rate, which is not horrible, but obviously we'd love to get that higher. And this is really important because the higher the numbers, the more valid the data is, the more representative that data is of our very heterogeneous and diverse population. And that's really important when we start thinking about how we can measure this disease, how we can measure treatments in this disease, and so on. So I won't cover all of the surveys because there are a lot, but in the short time we have, I'll just go over a couple here. And I know that nobody likes to look at these big bar graphs, but I'll walk you through them, and you don't have to look at everything here. But one of the first surveys that everybody takes is this head-to-toe survey. And this is a comprehensive survey that covers symptoms in all parts of the body or domains. So for here, we had 92 respondents out of the 125 that answered this survey. So the dark bar represents how many people said yes, and then the yellow, or I don't know if it's showing up as yellow or orange, that's the number of people who said no to that domain. And so you can see, this should come as no surprise that many of the respondents said that their symptoms have to do with the brain and the nervous system, right? And we know that this CACNA1A is a gene that's important for the brain. That makes sense. But you can also see that developmental issues, eyes and vision are in there as well. We see digestive issues, behavior and psychiatric issues, sleep. Those round out some of the top 10. You can see at the very bottom, things like cancer and blood and bleeding. Those are pretty rare in this community. So if we break the symptoms down further in that brain and nervous system domain, again, these should come as no surprise, but what are the top 3 symptoms that are reported in these domains? You can see that there's coordination issues, which is ataxia. We see that hypotonia, or our kids having that low tone, which makes them a little more, flimsy and loose. Does not mean that they're weaker. It just means that their muscle tone is a little is lower. And then cognitive impairment, which we know is so important for so many aspects of our lives. You still see things like seizures, abnormal EEGs, headaches, and migraines. So these are pretty much line up with what we see in the published literature. Now, something that you don't really see in the published literature is sleep. And Dr. Josh Rotenberg 2 years ago gave a great talk about some of these sleep disorders that are found in neurodevelopmental diseases, especially in CACNA1A related disorders. So again, you don't read much about it, but it's actually it's a big concern in our community. And so what you're looking at here is a graph showing the sleep disturbance scores from the Children's Sleep Habit Questionnaire. And this survey looks at different aspects of sleep in school-aged children. So this is really just for the pediatric population. So it looks at things like bedtime routines, bedtime behavior, sleep onset, night waking, sleep duration, and so on. And so, on the axis here, you can see the number of patients that reported the score, and then down here you can see the range of the scores. So we had ranges from 33 all the way up to 73. And the higher the score, right, the more likely that this child has some sort of underlying sleep disorder based on the questionnaire. And so we had 44 respondents, and the average score, as you see here, is a 46. Okay, so what does that mean, right? Like, why is that significant? Well, it's significant because based on these results, anyone who scores 41 or has a score of 41 or higher might have a sleep disorder. And as you can see, 61% of our respondents had a score of higher than 41. So I think that's a pretty significant indication there that sleep is an issue, and that is something that we should also be taking a look at. Something that you don't really think about either, or maybe hear about, is behavior. And the reason I'm also taking sleep and behavior is because we also have another disease. We have a disease concept model study that captures the burden of the disease on families and on individuals. And surprisingly, or maybe not surprisingly to some of you, sleep and behavior came up. So I was curious to see what the RARE-X data looked like in terms of the behavior domain. So we had 76 respondents, so a better number there. And you can see that while it's not really predominant, we still see things like temper tantrums and about 30%, unusual eye gaze, impulsivity. I'm sure some of you are used to just, these individuals not thinking right away and doing something, and sometimes that can be dangerous, right? And I also want to emphasize that, anxiety, hyperactivity, aggressive, violent behavior, when you see these things, it can be really alarming, right? Especially in young children, and you don't know how that's going to progress. And even though it's only a small percentage of this population right now, it's still, it's an additive effect because you're having all of these behavior symptoms on top of the other symptoms that are in the nervous system and the other parts of your body as well. And that kind of brings me back to then, again, that burden of illness. And I really want to emphasize, we are not calling our children or ourselves or our loved ones a burden, right? This is the burden that is placed upon them and us as caregivers and families because of that disease. And so something that RARE-X has kicked off is this burden of illness initiative. And it involves several surveys that really look at how the disease impacts those who are living with it. So again, a lot of numbers here, a lot of things to look at. I will orient you here. So this was a list of different symptoms, and the respondents were asked to rate each of these symptoms depending on how much they impact daily activities. And so the green is showing you a very high impact. The dark blue is showing you high impact. The light blue is a moderate impact. The orange is low impact, and the red is no impact at all. So again, I'm not going to go through everything. We only had 27 respondents, so obviously let's get those numbers up. But here's what we see so far. So you can see at the top here, I don't even know why I keep looking at that way. We have, I cannot read that at all. Excuse me. Developmental delay, communication, things like that are at the very top. And ataxia as well, these top threes. And you see that feeding issues, feeding tube, psychiatric illnesses, and hearing, few are reporting that that's an impact. But I think because we only have 29 people that responded, these are not representative of the true numbers. So we would really love if everyone could help us think about that. Another question that was asked based on these symptoms is that which daily activities are the most impacted? Because it's not only important to understand what the symptoms are, but again, how are they impacting the families around us? And so you can see at the very top, again, the numbers, the colors are changing, but the dark blue, unable to do it all, the blue, severe challenge, the light blue, moderate challenge, the yellow, minor challenge, and then the red is that there's no challenge. But you can see things like self-care, being able to attend school or hold a job, participating in social activities, not being tired. I mean, these are things that many of us take for granted, but we have to remember how hard it is for these individuals, because it takes so much more brain power. It takes just so much more energy to do things that we can do. And so it's important to understand how these symptoms are impacting everyone. And it's also really important information, because this is what we're going to tell the FDA when we have these listening sessions. We're going to bring in clinicians, we're going to bring in families, and have everyone talk about what is missing in our community in terms of treatments, and what are those priority treatments. But I also want to emphasize that we need to get these numbers up. And so RareX told us if we can get 61 respondents, we will be included in a burden of illness publication that they're going to be writing, and we will be helping them write that as well. But again, once this is published, it's available for researchers and clinicians and the FDA to see, and we can't move forward without any of that. All right, so just a quick recap. What have we learned? We've learned that, again, main symptoms are reported in the brain and nervous system domain. And the top nervous system domains are, again, the ataxia, hypotonia, and cognitive impairment. We also have underreported symptoms in these domains, such as sleep and behavior. And all of that, right, additive effect, all of that impacts these daily activities like going to school, holding a job, brushing your teeth, getting dressed. That really does prevent these individuals from having that quality of life that they deserve. And all of this, again, is going to be used to help develop treatments, not just to improve the quality of life, right? That's what we're going to aim for right now, but hopefully one day down the line, a cure. And I know that's going to mean something different for a lot of you out there. But we are working, again, to get those treatments that will improve the quality of life for now, and then one day a cure. And a big part of it, too, is understanding and defining what we call these biomarkers or outcome measures, which, again, sound like these weird words that we keep saying over and over again. But think of these as parameters, right? How do we measure this disease, right? And how do we measure change? Because that's the most important thing. We could have the best drug in the world, but if we can't show the FDA that there's a significant change from before the drug and after, nothing is going to get approved. And so we really need to focus on these biomarkers and outcome measures that can also be taken from the data that we collect here. Okay, so you want to be counted. All you need to do is go to our website. You can either visit this webpage here, which I know nobody really goes to. So here we go. There's a QR code. Go ahead and scan that if you don't want to type the whole web page in. And I believe we will also have information how to sign up for the RareX study out where registration is. And of course, thank you to RareX for gathering all this data, and to all of you, you wonderful CACNA1A families, for giving us this opportunity to find out this information directly from you. All right, thank you. Wendy? Okay, so I'm going to introduce everybody next to Dr. Wendy Chung at she was at Columbia University. She's now at Boston Children's, and the natural history study has moved over with her, and she's going to give us an update.
Thanks, everyone. Hopefully you can see me and hear me. This is really showing you back your own data, and hopefully in a way that makes sense. I'm going to go relatively quickly through this because it actually is larger but consistent with the RareX study. And so hopefully this all makes sense. So I can't say enough how important this is in terms of clinical trial readiness. As some of you know me, I'm a geneticist for rare genetic diseases. And we work on many of them. And I can say for some other conditions we're working on, we either have or are moving into clinical trials. And so we know what it takes when it gets there. And it is really critical to be ready because you are in some ways competing against other groups, and being able to be prepared and show your numbers is important. And we can talk about this a little bit more. The natural history study we've tried to make as easy as possible for people to participate, and we've taken very much to heart your comments in terms of how to do that. And the numbers I'm going to show you today, and I know we've had a lot of discussion already, I am focused only on the variants that are pathogenic or likely pathogenic to give a very clear answer in terms of what we're seeing. For those of you who have participated that do have variants of uncertain significance, we have your data. We could easily run similar sort of graphs and show the data, including them, but we wanted to be certain of the information that we presented. This is a living registry, so we are looking at people over time, and that over time is critical because things do change over time, either as seizures are or not under control, as people are going through different life stages, puberty, and then progressing into adulthood. And so this is critical to understand how those changes occur. And I can say with other clinical trials, that's been critical to know without drug what happens over the natural history, over that natural aging process. So let me show you who we have and who we don't have, importantly. So this is the individuals with those pathogenic or likely pathogenic variants, approximately 100 individuals. It is skewed towards younger individuals. And so this is a distribution on the x-axis by age in deciles or by 10-year periods. As you can see, most individuals are less than age 10 and certainly less than age 20. We do have a few adults who are in the study, but this is not because there aren't older people out there with CACNA1A. They may not be accessing genetic testing and or they may not be participating in research, but we are skewed. Interestingly enough, we're skewed a little bit more female than male, although in theory we should see equal numbers. And that's an interesting question, but may just be a small sample size. In terms of where people are, they really are global, although, and this may be reflective of who's seen this as the opportunity as being a little bit more North American based, but we do have individuals from around the world. And based on our other research studies, we should be relatively equal opportunity around the world. There really shouldn't be a bias in terms of parts of the world that are over or underrepresented. You've heard a lot already in terms of all the genetic discussions, so I'm not going to go through it in too much detail, but most of the variants are missense variants. We actually discussed with the family group leaders ahead of this whether to try and classify the data that I'm showing you today by either predicted loss or gain of function. We ultimately decided not to do it that way, although I have run some of those analyses, and consistent with some of the earlier comments, I'd agree for this channel and other channels that gain of function tend to be overall in the aggregate more severe in terms of associated clinical features. But on the other hand, every variant to extent is their own variant. For most, but not all the variants in the cohort, these are so-called de novo or new in the individual. Of course, many people didn't have parental testing and so were unknown, but and those, of course, could be de novo. I'm not going to go through in detail all of these. This was really meant to be more for the researchers. I will point out, because some people have been calling out variants this morning, this actual nomenclature about which variant depends on something called the transcript ID, and there may be slight moving over one amino acid versus another. But just to say that most individuals are onesies or twosies, meaning that in most cases, there aren't large numbers of individuals with the same variant. There are some exceptions. There are also, as you've heard, certain types of variants that we tend to lump together as being ones that probably destroy the function in terms of just not having protein produced. And to a certain extent, we may be able to aggregate those together simplistically, and I've shown those over here on the right. And then for those of you who are visual people, this is what you've seen to a certain extent in terms of the different domains of the channel and where those variants are located. And I've color-coded these in terms of gain or loss of function, gain in red, loss in blue, by prediction. So these are not always based on actual experimental data. The numbers in the circles, inside the circles, are the number of individuals that have the same variant. These individuals are not related to each other, so these occur independently. And as you'll see, a few of them are recurrent or happen in more than one person, but that's more at least in the data that we have, not commonly true for most variants. Okay, let me go relatively quickly to the clinical characteristics. I'll say that many of you were astute observers as parents and noticed things relatively early, even in the newborn period. Difficulty with feeding, your baby being a little bit more sleepy or less responsive, a little bit more hypotonic or loose in terms of tone. And so those things, in retrospect, you did report. With this, though, largely a neurological condition, but to the extent the brain affects the way the body functions and moves, we see manifestations in multiple parts of the body, the eyes, the arms, the legs, obviously in terms of thinking and seizures as well. In many cases, one individual has more than one manifestation, and this may even vary both in terms of severity or when they occur over the life course. And so some things may develop over time. Movement problems with coordination, the fluidity of movements, very commonly affected. That can be called ataxia by your neurologist or your doctor. And then other things that we see in terms of headaches, migraine headaches, hemiplegic migraines, or alternating hemiplegia. So those oftentimes seen and categorized as such by neurologists in terms of a diagnosis they might have given your child or you. With that, in addition to problems with coordination or ataxia or movement problems, some individuals have visible tremors that we can see, or problems in terms of dystonia. Within this, I'm showing on the right the age of onset so that you have some sense based on the age of your child, whether or not you still might be seeing those things occur. And you can see the range in the parentheses on the right that for some of those, they may not be present within the first year of life, as an example, even though they might, on average, start early, they might still, for some individuals, be recognized later in life. In terms of other things that we see, high pain tolerance is quite common. Problems with seizures, for those of you who have that, certainly one of the things that's most impactful. And if seizures are under control, can be very beneficial. And if they're not, can lead to other problems. Within this, as again shown on the lower right, is when we oftentimes see these starting. And for most individuals, we see these types of symptoms starting quite early. For those with seizures, there are many different seizure types, and any one individual can have more than one type of seizure, but status epilepticus is something that's quite concerning and unfortunately quite common. Other things, so-called absence or petit mal seizures, where someone just looks kind of dazed and out of it, also quite common, as well as various different types of seizures where you see movement, body movement, either generalized tonic-clonic or simple or complex partial seizures. But seizures in general can unfortunately be intractable in many individuals, and even to the point of daily seizures or monthly seizures. So again, a focus for many individuals in terms of potentially a primary outcome measure for some clinical trials. In terms of things that you've seen on brain MRIs, we can, not everyone has something, but certainly these can be seen, and in particular in the cerebellum or cerebrum. With these, as we're seeing this, these may have been normal in the past in about a quarter of individuals, and something I doubt we'll use for an outcome measure in terms of clinical trials, but something that can be seen. Eye symptoms are important in terms of both how to accurately see the world around you from a safety point of view, from a learning point of view. Some of these things treatable, some of them more difficult for treatment, but nystagmus or movement abnormalities, something that can be seen, as well as issues in terms of just literally things that can be treated with glasses, either if someone has problems with either being seeing close or far away, but also problems that individuals can have with cortical visual impairment, thankfully less frequently, but something like cortical visual impairment not treated with glasses. In general, development is an issue, and this is what I would say is pervasive developmental delay for many individuals. That is problems in terms of both learning to move their body, either in terms of big movements or small or fine movements with the hands. Also, problems in terms of expressive language, whether it's actual spoken language or other ways of being able to express oneself. And with seizures in particular, if they're not under control, sometimes leading to regression or loss of skills or stagnation. So obviously difficult. In terms of how delayed these are, these can be a range, but just to show you on average, the age at which a first milestone was attained. Individuals largely are making forward progress and forward momentum, although at a slower rate. But we do have individuals who are, as an example, not achieving certain milestones, such as spoken language. There are other means of communication, and so certainly I think technology is helping us in that, in addition to spoken language. As I said, regression, I think, is one of the things that worries us most. For some individuals, it's temporary and some of these skills come back. The bigger concern is for those that are permanent, and there's a fair number of individuals who experience permanent regression or loss of skills. This was mentioned earlier, but behavioral symptoms also can be an issue. So we see about 20% of individuals with autism. Any one person can have more than one of these symptoms. Problems with attention, anxiety can often be an issue, especially when there's disruption of a routine. Some other behavioral diagnoses as well. And for individuals, as I said, these can also change over the life course, that during puberty, for instance, there may be different diagnoses, behavioral diagnoses, than for young children. In addition, there's a connection between the brain and the gut. And with that, we've commonly see not just with CACNA1A, but also other neurogenetic conditions, issues that can be constipation, diarrhea, reflux, but these come up frequently, and then in part due to the low muscle tone, problems with scoliosis or curvature of the spine, oftentimes during adolescence, so something to keep an eye on. I'm getting close to the end, but I wanted to show you some of what was working and not working in terms of specific medications. So I've broken these down by by what the medication was used for. This is shown consistently across these next set of slides. Those in green were people who responded well using the medication. Those in orange were people who didn't get the desired response with the medication. And to some extent for epilepsy, has been a trial and error process. Some individuals have required more than one medication, or even beyond a medication, other modalities of treatment. Valproic acid, just by frequency, was the one that empirically had the best response rate. And I'm not saying that valproic acid will work for everyone, because it obviously didn't here. But different medications have been used by different individuals with different rates of success. There have been some side effects with medications, different people with different side effects, and certain individuals, just it didn't completely treat the epilepsy. In certain individuals, they got lethargic and were sleepy, and with that, less able to learn, and may had other behavioral issues as well. For ataxia, a couple medications of note, acetazolamide used quite frequently with responders, more likely than not, although still not everyone. Acetyleucine, not used by very many, but for 2 individuals, it did work well. For migraine headaches, acetazolamide, verapamil, topiramax for AP, things that were used with variable frequency and variable success. Again, in terms of this, you've already been hearing about some of this, and I'm sure we'll be talking more about it at the meeting. So just to be able to wrap up and say, again, most of the variants are missense variants. I do think over time, I hope we'll have some better correlations between individual variants and then trajectories, and most importantly, response to therapy. It will be interesting to see how we design clinical trials, keeping in mind that heterogeneity. And when we've done this for other conditions, the best way to account for that has been to have very good baseline and run-in data for each person, so that each person could effectively serve as their own control. Hence the reason why this phase right now, before we're in clinical trials, is so incredibly important. And again, most of the features focused on those in the brain, but also outside the brain with the visual symptom, the GI symptom. So for those who've been part of this at Columbia, as was said, we're continuing uninterrupted at Boston Children's Hospital. The same individuals ported over in terms of their data and ability to use their data. And if anyone is wanting to join a SEND study at children's.harvard.edu is the email. And I just wanted to put a shout out. Jessica Waxler and Emily are in the audience with you there. They're there through the meeting, and we'll have a table this afternoon so that people can sign up with them and get them onboarded or let them know where they left off. So I'll stop there. Thanks.
Thank you so much, Wendy. And our next presentation is a recorded presentation from Terry Jo, but she will be live after the presentation, correct? To join us for a question and answer session.
Okay, good morning. This is Terry Jo Bichelle. I am the CEO of Combined Brain. And I'm going to talk today about clinical trial readiness for the CACNA1A field. Thank you so much for letting me present. So Combined Brain is a consortium of over 100 member organizations, and each one of these organizations represents a rare genetic neurodevelopmental disorder. CACNA1A is one of these. This consortium, all these different groups work together to try to pool resources so that we can all get ready for clinical trials together. It's an exercise in collaboration and efficiency. So the slide I always show about the path to drug development for a rare neurogenetic disease follows this particular trajectory. First, you identify the gene, come up with your models, collect natural history, try to figure out what the gene mutation is doing that's causing the disease. At roundabout then, somebody gets an idea, ding, for how they can do a potential treatment. But you still have a long way to go before you can get that drug or that treatment into clinical trials. You've got to do conceptual models, which tell you exactly what people care about who are struggling with the disorder or caring for people with the disorder. You need to develop biomarkers. You need to come up with outcome measures, which are the best way to measure whether a treatment is working. You have to do some safety tests to make sure your drug is safe. Then you design your clinical trials and you finally apply to the FDA. So everything above this line, I usually say is really the responsibility in many ways of the foundations. Everything below this line is usually the responsibility of academia and industry. But no one above the line can exist with anyone below the line. It's a back and forth, back and forth, back and forth all the time. So what Combined Brain does is we try to fill in all the blanks in that whole line about all the different tools and resources that are needed to get from identifying the gene to an approved treatment. And so we have a lot of different programs. We have programs to diagnose infants with genetic disorders, programs to help groups apply for rodent models. We have a biorepository. We have scientists who work for Combined Brain who can actually work for each individual member organization. We help with natural history and ICD-10 codes. We come up with outcome measures for different symptoms that we know are important to families because we've asked them what's important to them. And then we also approach the FDA. But today I'm really going to concentrate on the biorepository and the biomarker programs that Combined Brain does. And let's see if it'll load my next slide. and it looks like it's having trouble. Let me see if I can get it. Well, if I can't get the next slide to load, I will start telling you about our projects. Okay. So in fact, the next slide is directly from the CACNA1A Foundation website. What it shows is the roadmap that the foundation has come up with, and you can see that it actually follows many of the same, it's seeking many of the same goals along the path. And all of these are really important for that path to drug development. So every one of these studies matters. And these are all studies that the CACNA1A Foundation has been endorsing. And that includes your contact registry, the natural history study under Dr. Wendy Chung, which I think you're going to hear about shortly. Rare X, a way for you to enter natural history information directly as a family. Citizen, a way for you to collect your medical records. Combined Brains Biorepository, which I'm really going to talk about a lot. A quality of life survey, which your foundation is sending out. All of these things are really crucial to drug development for this disorder. So you all have been amazing and very active in collecting samples for the biorepository. Thank you. On behalf of Combined Brain and also on behalf of your disorder, you have collected all of these samples from each one of these lines is a different donor. And this column on the right tells you all the different types of samples that that person has put in the biorepository. Anything from plasma to urine to whole blood, and even to stem cell lines. And all of these different types of samples are used to do research for biomarkers. And what are biomarkers? Biomarkers are something that can be measured, so not an opinion, but something that can actually be measured, usually in blood or body fluids, but can also be something like an EEG, which I will mention in a minute. And ideally, you've also collected enough natural history that if you measure something, let's say in plasma, you can see that it might be increased at the same time that a person's cognitive abilities are increased, or it might be decreased at the same time a person's cognitive abilities are increased. In other words, that biomarker is linked to something that you can actually experience in the person. So the biomarker is a way to measure whether or not the symptoms will be improved. And yet the biomarker is usually a much more objective way to measure progress, even though what we might care about more is is the person cognitively improving, or are their seizures getting better, or are their motor skills getting better, something like that. Sometimes those changes are really, really hard to measure in the short term. If you can imagine someone increasing their learning and memory, they're going to have to be able to learn and memorize things before you can be able to measure an increase in that learning and memory. But if the biomarker in the blood, some protein, some chemical, some EEG pattern, if that biomarker is improving, then that gives us the time and the patience to wait while the learning and memory improves, and we're able gradually to measure an improvement in, for example, cognition or seizures or whatever else we are trying to track. So what we've come up with at Combined Brain is a way to share these biomarkers, because they're going to be important to everyone in the field. You might have one company looking at one particular treatment, another company looking at another treatment. And so you may actually it's going to help them if they come up with the same biomarker that they can share. Same thing with helping one patient advocacy group, maybe a different disorder. They may be looking at the same biomarker, even though they may have a totally different treatment or a totally different disorder. So what we've come up with is this plan of doing pre-competitive biomarker projects. So all the different groups in Combined Brain are doing their donations of blood and plasma and all the other things. And then we're sending them in together to get analyzed, and the costs are covered by a whole group rather than just one patient advocacy group or just one industry partner. So in fact, our first biomarker project, your samples have already been sent, and we're now waiting for the results to come back, and they should come back any day now. They're actually due in July. We'll still need to analyze those results, and we'll also need to see if those results are correlated to the measures of severity, such as seizures or motor or cognition. But you all included 28 samples in the first pre-competitive biomarker project, which is measuring proteomics in plasma. Those are different levels of protein. And of those, 18 of your samples were from patients and 10 were from controls. So this is very exciting, and hopefully we will be able to see something that shows a change or something that is different between the patients and the controls, and ideally something that is different between the patients that are less severely affected versus the patients that are more severely affected. And then we have a second pre-competitive biomarker project coming up, which is metabolomics. Those results are not going to be due for a couple months. And we have a third one, which is methylomics. All those are different ways of measuring proteins or changes in the genes in the plasma. So why are biomarkers so important? Well, one of the reasons is what I mentioned before, that you sometimes, or if you have a great biomarker, you can sometimes see a change in the biomarker before you can even see a change in the person. And this has been really true for Hunter syndrome. And Hunter syndrome right now is really trying hard to work with the FDA and work with all the different groups who are working on this similar type of disorder to use heparan sulfate, which is a particular something that you can measure in blood or cerebrospinal fluid, and it is elevated when the disorder is not treated. And as the disorder is treated, this level of heparan sulfate is reduced. And so what you can see with this particular schematic, which sort of explains how you can measure the level of heparan sulfate and almost predict the symptoms of the disorder. So when the heparan sulfate is pretty low, is below this line here, the person is not experiencing these symptoms of the disorder. Once it hits this line here, they start to have neuroinflammation. Once the heparan sulfate goes up to this point, they start to actually have neurodegeneration. So nerve cells are starting to recede. And once they hit this point here with the level of heparan sulfate, they're starting to have what you can actually see, which is neurocognitive impairment. And then once they get up to this high level, you start to have actually visible neuronal cell death. So the brain starts to shrink. So what you can see is that it's really important to start treatment early. And if you can start treatment way down here when the heparan sulfate levels are really low, and if you can keep that heparan sulfate level very low, you might be able to prevent all of these later stage symptoms and problems. There's another biomarker which is really exciting for Rett syndrome, and this actually shows how what you measure in humans needs to or can be correlated with what you can measure in other animal models, for example, rats or mice. So this is another study that was done in Rett syndrome, where they measured in the cerebrospinal fluid proteomics also, and what they found was that across all three models, mouse, rat, and human, they could find two different gene expressions or protein expressions, ApoA1 and VGF, which would be elevated during Rett syndrome and when the disorder was worse. And so the idea is that with this biomarker, you could actually do experiments in the mice and rats, for example, with gene therapy, and see if you can reduce that ApoA1 and the VGF. And if you could, then that might be something worth trying in human patients. And then you can look to see if in their cerebrospinal fluid, you could also be measuring reductions in ApoA1 and VGF, and then you might know that your treatment was starting to take hold. Another biomarker project that just came out a couple of weeks ago is a plasma proteomics project, which has actually identified 7 different biomarkers together. So if you look at all of those 7 together, you can actually predict Parkinson's disease up to 7 years before any symptoms start. So this is a complicated biomarker because you've got to measure 7 different proteins at the same time and look at their relation to each other. Some will be up and some will be down. But it's looking to be a very accurate predictor of Parkinson's disease, which will be amazing to have a blood test of Parkinson's and maybe be able to treat it before any symptoms start, which has not been possible before because we haven't been able to predict when Parkinson's is going to happen. And the way they did that, it's the same way that we're working on looking at the proteomics in the CACNA1A samples. The first thing they did was turn in a few samples. So they turned in 10 to 20 samples, just like you all did. And then they looked at every protein they could possibly look at. And then they found a certain subset of those proteins that seemed to be off in the samples of people who had Parkinson's. And then they got more samples of people with Parkinson's to measure those. And then they brought it down to the seven proteins that were off in relationship to each other. And at that point, they needed to bring in a lot more samples to see if those 7 proteins were actually really a biomarker for Parkinson's. And that's what they saw. So as you can see, we're just getting started with these biomarkers. We're in the middle of the very first experiment. What that means is we're going to ask you for even more samples once we come up with results of this experiment to be able to move to the next one and start to understand what biomarkers are going to be important in CACNA1A. And that goes along with another effort that we're doing, which we're not quite ready to launch yet, which is the Combined Brain EEG Bank. And for that, we need to collect a lot of EEGs from people with CACNA1A syndrome. And that means it could be EEGs for people who have seizures, EEGs for people who do not have seizures, EEGs for at one time point, and preferably even EEGs longitudinally. So if you had 5 EEGs in your life, we would like to collect all 5 EEGs. If you had EEGs, different kinds of EEGs from different places, one that was a 20-minute, one that was 24 hours, we would like to collect all of those EEGs. We're going to be storing them in one big EEG bank, which is a lot like our biobank, only it's an electronic resource. And those EEGs will be made available in raw form to any researcher who needs to look at them. And that could be a clinician in a hospital who's working on an academic project with the EEGs, or it could be an industry partner who's starting to research CACNA1A because they'd like to be able to treat it. And then after we collect all the EEGs you've already done, we'd also like to start collecting EEGs at your house in the future. So that would be phase 2, and that's going to come up in a couple of years. So why is this important? Well, I can tell you from my personal experience as the mother of a son with Angelman syndrome. I worked on natural history studies in Angelman for many years. I worked alongside and under Dr. Lynn Bird, who, and in that natural history study, which was held in 6 different cities, and the overarching PI was Art Baudet, we collected hundreds of EEGs. And we collected enough EEGs, and also over time on the same person over and over again, we could actually get those EEGs analyzed. And what we were able to find out is that we could show the phenotype of Angelman syndrome. We could show a signature, what we call an EEG signature, a certain pattern in the EEG that was always there with Angelman. And it was so predictable that we could actually tell the difference between genotypes in Angelman syndrome. That was a big deal because some people have deletion, some people have a point mutation, some have other imprinting center mutations. We could tell the difference from the EEG, whether the person had deletion or non-deletion. And then it got even better. We had to collect even more EEGs and analyze them even more. But this gentleman here, Jorg Hipp, who works for Roche, Hoffman LaRoche Corporation, further analyzed those EEGs in detail and also correlated those to the Bailey, which is a test of cognition and development. And he was able to show that that EEG score correlated with how the child scored on cognition, motor skills, and communication. Those are the things that families care about. And these are kids with or without seizures. The seizures were not the crucial part. It was the EEG signature. So what that means is that if we treat Angelman syndrome with gene therapy or an ASO, and we correct that EEG pattern, hopefully that will correlate to these other measures which we all care about, how our child moves and thinks and learns and communicates. And in fact, at this moment, there are antisense oligonucleotides in clinical trial, and the EEG pattern is now being analyzed to see if it will correlate with those measures of development. So we want that for CACNA1A. We want that for your disorder as well. And so what we need natural history, which is crucial, and you're collecting it through Dr. Chung, through Citizen, through RareX, and maybe through Combined Brain as well, or maybe only by, we can also sign data agreements with those other natural history data collections, so you don't have to fill out even more surveys. But even if you do have to fill out a survey, I think filling out surveys is a small price to pay for trying to get a treatment for your child. And so that is my presentation for today. I'm really eager to hear from Dr. Chung and to participate in the panel. And thank you very much.
Well, you actually missed Dr. Chung, Terry Jo, but that's okay. She was first. Thank you, everybody, for explaining all of this to us. And I think that what people need to understand is that while our scientists are busy working on disease-modifying and symptomatic therapies, we've been collecting data, and so I hope you understand a little bit about the importance of that data and why we're doing that. So, anyone have any questions? Brennan? Oh, okay.
Yeah, thanks. So Lisa, we fill out these surveys, but we forget which ones we filled out and which one we haven't. How can we figure out what we've done and what we haven't done so that we do the ones we haven't done?
That's a really good question, and that is a problem. We understand that's a problem. You should be getting emails, regular emails from RareX, and an annual update from the Ascend study asking you to update everything. But just in case, we realize that this is a problem, and there are so many studies, and unfortunately, there are going to be more studies because this data, as we learn more, we need to collect more data. So we are starting something that I mentioned in my talk earlier called Cure Club. And so that's being put together, and there's some brochures outside with a link that you can sign up. And as I mentioned, Shanna is organizing that. And it will be, just a way for you to check off which ones you've done and which ones you haven't, and a touch point so that you can say, hey, have I done this or have I not done this? For those of you who are here today, we do have people from Dr. Chung's lab who can answer any questions about where you are in that process. And Deb, right? And Deb will be out there overseeing RareX, I believe, correct? Or just everything in general. So, and there's Deb.
I'll just explain Cure Club. It's awesome.
I noticed in Dr. Bichel's talk about this biomarker repository that Combined Brain are putting together. And one of the really interesting sort of collections that they're doing is for iPSC cells. I presume these will be hopefully used to be almost as Dr. Bichel was suggesting, to be almost characterizing these iPSC cell lines directly taken from potential patients to be characterized in any number of manner of ways. Is that how they'll be used?
Yes. So, right. So these iPSCs, right, are going to be differentiated or reprogrammed into different neurons. And so it is a way that we can characterize what some of these variants are doing. So you can look at things, you can grow these neurons, and then you can just look at things really, from a high-level aspect. Are the neurons longer than our wild type, or neurons that don't have the mutation, right? Are they longer?
Explain wild type, what wild type means to.
Okay, yes, a wild type is simply a cell that doesn't have the mutation, so it's got a healthy copy of the gene. So you can compare it against wild type. Are the cells long, or the neurons longer? Are they shorter? And then you can do things like electrophysiology experiments, sort of like what, similar to what Jen explained with those hex cells. You could do the same thing in these neurons. You can look to see, are they firing at different rates, right? Is that neurotransmission or their communication, is that better or is that worse? So that's how researchers can use these iPSC lines. And another important point that we want to bring up too is that we always have an open call to collect blood samples for those iPSCs, but we may not always make we always collect blood to make those iPSC lines, but we may not always make those initially because they do cost money. And so we have this collection, but if a researcher is interested in having one of those lines developed, then, we can talk to them about it. But it's really trying to find the variants that have a very robust readout, something that we can measure that change in. And so, again, these can be used for many different research purposes. And because they're in the biorepository, they are available for researchers. You just have to contact Combined Brain, or you can contact us directly.
So the only other thing I was going to add is that we Combined Brain does not collect samples in countries other than the United States. But if a person travels to the United States, they can give a sample while they're in the U.S. Or I believe if they travel to Dr. Chung's study, they could give a sample there and perhaps transferred into Combined Brain. Vice versa, if we collect a sample, we can transfer it in to another study as well. And in fact, if they collect a sample overseas, and we can get a contract with that academic researcher or clinician, we can transfer that sample in. So it's possible, it's just complicated.
Okay. Any other questions? Okay. Well, I'll just sum up by saying that you know, I just want to clarify one thing, what a biomarker is, because I think it's sort of important. And when we're doing these clinical trials and when we're doing the studies out in the hallway, we're looking at what, and I may be wrong, so somebody help me, but an endpoint is something that you can visually see. So is your kid, how are they walking? You know, are their hands, are they having a tremor? Are they having a seizure? And a biomarker is really something that we measure that you can't see. So it's in your blood or it's in your brain, and we need an MRI, or we need to actually look at the EEG. And these are used by the FDA to measure if a treatment is working or not. And so again, just to kind of hone in on the fact that we ask our community to fill out all of these surveys, and we know that they're tedious and they take a lot of time, but you can start to see the value when you look at other diseases like Parkinson's and cystic fibrosis, and when you have a large community, what can happen. And so we actually are a pretty large community and a growing community as more and more kids are diagnosed, and as genetic testing becomes more widely available. So we need to use our voices to tell people about how important it is.
Sorry, Lisa, I just realized I didn't ask my question, and I wanted to make sure this was, people heard it directly from Dr. Chung, because it comes up a lot in our community's Facebook group. People contact the foundation all the time about this, and it's about the VUS question and whether an individual who has a VUS can enroll in the natural history study, and if so, where that data is, what happens when the variant is reclassified. Does the family need to get back in touch with you to let you know? Are you guys monitoring that? And are there certain VUSs that you're excluding from enrollment altogether?
So I hope you can hear me. We assess the variant, and VUSs are welcome. If we determine that it's really not a VUS, but likely benign, and that rarely happens, but sometimes happens, we let you know, because in some cases I've helped people get to their right diagnosis, not their wrong diagnosis, and we want you to get to your right diagnosis. We are reassessing VUSs over time. We actually have a large publication in Genetics and Medicine describing exactly how we do that. And when we do that, those data then move into the ones that are pathogenic and likely pathogenic and get incorporated into the total set. So as you've heard earlier, we realize this is dynamic, and we keep evolving as the knowledge evolves.
So everyone sign up, VUSs included. Thank you.
Thank you so much.